Hello everyone. Today we will discuss about the beta oxidation of the fatty acid, especially of the palmitic acid, that is 16 carbon atom unit. Now, first of all, what is beta oxidation of fatty acid? As the name says, beta oxidation, that means the oxidation and the splitting of the two carbon unit mainly occurs at the beta carbon atom position, means somewhere over here. It will be breakdown. Now, it has mainly two steps, preparatory steps and the step for the beta oxidation. Now, first of all, preparatory step for beta oxidation. Now, in this, as you can see, this is the coenzyme A structure. Okay. Now, it has mainly this pantothenic acid, means vitamin B5, and this is the mercaptoethanolamine. It has SS group, which will actually form thiazide bond with the acyl-CoA. What is acyl-CoA? Just remember, just know the difference between the acyl-CoA and acetyl-CoA. Acetyl-CoA, that means the connection of the acetic acid along with the coenzyme. While acyl-CoA, that means the connection of any fatty acid along with the coenzyme. So over here, just remember, beta mercaptoethanolamine will form thiazide bond with the acyl-CoA. So this is the structure of the coenzyme A. Very important structure for this beta oxidation purpose. Now it has mainly four preparatory steps. Okay. So first of all, step 1. Activation of the fatty acid. Now just remember, this process actually occurs into the cytoplasm. Only this step. Rest all steps will occur in the mitochondrial matrix. Okay, this is the first step for the beta oxidation and occurs in the cytoplasm. Now over here, the fatty acid actually activated into fatty acyl-CoA by thiokinase or the fatty acyl-CoA synthetase with the help of one molecule of ATP. Over here, just remember, two high energy bond are used, so that's why ATP is converted into AMP plus PPI. Okay. Just remember, it occurs in the cytoplasm. And we have mainly three types of fatty acid, if you know: long chain, medium chain and short chain. Now this thiokinase mainly acts on the long chain fatty acid, while thiophorase acts in the short chain fatty acid. Now, step 2 is the role of carnitine. Now first of all, you may know that the long chain fatty acid only needs to be transported inside the mitochondria with the help of all these transporters. Medium and short chain fatty acid can easily pass into the mitochondrial matrix easily without any help of the energy or transporter, but long chain requires. So for this, we will do the rest 3 steps of preparation, that is the role of the carnitine. Now what is carnitine first of all? It is the beta hydroxy gamma trimethyl ammonium butyrate. Just remember, beta hydroxy gamma TMAB, trimethyl ammonium butyrate. Now what it will do? Now this carnitine, I told you before that the beta oxidation occurs in the mitochondria, and so transport of the fatty acid through the mitochondrial membrane is essential. So the long chain fatty acyl-CoA cannot pass through the inner mitochondrial membrane, and therefore transport the carnitine is involved in transfer of the fatty acid. So this is a transporter. Carnitine is actually a transporter, and it is mainly synthesized by lysine and methionine in the kidney and the liver. So this carnitine is actually long chain fatty acyl-CoA fatty acids transporter. Now next is the carnitine acyltransferase 1. Now in this step 3, what happens is, this carnitine acyltransferase will be present in the cytosolic part of the mitochondrial membrane. Now what it will do? It will form the acylcarnitine. Now this, just remember that the acyl-CoA or the fatty acid, out of which this CoA can easily transferred into the mitochondrial matrix, but this acyl needs some transporter. So what CAT1 will do, carnitine acyltransferase 1 or the CAT1 will do, it will just connect the acyl group of acyl-CoA to the hydroxyl group, OH group of the carnitine, and form acylcarnitine. Okay. Now next, last step for preparation is the translocase. What is this? Now this translocase will actually transfer the acylcarnitine into the mitochondrial matrix. Okay, so it will just transfer this into this. But what about the acylcarnitine? Now this acylcarnitine, after entering into the mitochondrial matrix, over here in the matrix part of the membrane, mitochondrial matrix part of the membrane, there will be CAT2. In the outside, there will be CAT1, Inside there will be Cat2. Now function of Cat2 is that this will break the acyl carnitine and will transfer the acyl group of this to again back to acyl CoA, and carnitine again reaches back to its original position for the next cycle. So we have 4 steps for preparation. Activation of fatty acids occur in the cytoplasm, carnitine role, carnitine acyltransferase 1, and the translocase and Cat2. Now clinical application of this first of all. Now over here I told you that the medium and the short chain don't require any kind of carnitine for transportation. They can easily transfer inside the mitochondrial matrix. Now there is one more thing. What if there is carnitine deficiency? So of course this mainly occurs in the infants, in the small babies or the preterm babies, in whom fatty acid oxidation is very required for the growth, right? But as because the carnitine is not present, of course if carnitine won't be present, so the transfer, this carnitine is actually the transporter of the this acyl group, right? But if this only is not present, so entry of this is not possible, and fatty acid oxidation or the breakdown is not possible, and the energy requirement is more because fatty acid doesn't breaks down in this, and energy required is high. So what will happen is the glucose is utilized more in the small kids, and that's why the small baby suffers from the hypoglycemia, because fatty acid is available but it is not breaking down. So of course they need energy, so body will use glucose only, so they will suffer from hypoglycemia. Now next is translocase deficiency. Now what happens is in this the translocase deficiency occurs. So what happens? It leads to defective metabolism of the long chain fatty acid, and in this condition the muscle cramps are precipitated by fasting, exercise and high fat diet. Now Cat1 deficiency. Now it is mainly inherited and affects only the liver, and Cat2 deficiency actually affects the skeletal muscle, and in severe condition it will affect the liver. So just remember, Cat1 affects the liver, while Cat2 in the primary stage will affect mainly the skeletal muscles and then the liver. Now what happens in this? In Cat1 actually results in the reduced fatty acid oxidation and ketogenesis with hypoglycemia. Okay, the ketogenesis occur with hypoglycemia. So means reduced fatty acid oxidation and ketogenesis with hypoglycemia, because the body will use more and more glucose only, so hypoglycemia occurs. And Cat2 deficiency mainly occurs, mainly causes the skeletal muscle atrophication. Now the sulfonylurea drugs. These are the 2 sulfonylurea drugs, glibenclamide and the tolbutamide. These 2 drugs are mainly used in the treatment of the diabetes mellitus 2. Just remember what is this? Diabetes mellitus 2 is actually occurs due to the deficiency or the lackage of the GLUT4 named glucose transporter. It is not because of the deficiency of the insulin, but because of the GLUT4 transporter which is not present in the skeletal muscle. So it mainly, now these sulfonylurea drugs are mainly used for treatment of diabetes mellitus 2. Just remember the name, glibenclamide and the tolbutamide. Now they mainly because of this, now this mainly used in the reduced beta oxidation and therefore glycemia by inhibition of the CPT1. So this was all about the preparative steps for the beta oxidation. We will talk about the main steps for the beta oxidation or the breakdown of the fatty acid in our next video. Hope you are well. Thank you so much.